Hello everyone, I'm Diyue Bu from Indiana University Bloomington. Here I'll present our paper, Haplotype-based membership inference from summary genomic data. I'll introduce our paper from these four aspects. First, let's look at the background of our research. Based on current human genomic data sharing policy, when the researcher asks about the allele information from a private human genomic database, the database could return either the allele frequency or the allele existence. For example, the researchers could ask about the information of allele A at position 1, 2, 3, 4 on chromosome 5, and the database could either answer 0.2 as allele frequency or true as the allele existence like Beacon database. The researchers have investigated ISMB-based membership inference on summary genomic data. Both consider the format of allele frequency and allele existence. They always used the hypothesis test to determine whether a genome carried by the individual is in the database or not. For the membership inference on frequency table, both Homer's attack and the paper by Wang et al. Investigate the difference between the distance of the individual to the target population and to the reference population. And for the membership inference on Beacon table, both the SB attack and the optimal attack compute the probability difference under the null hypothesis and the alternative hypothesis. Here we propose the haplotype-based membership inference to evaluate if a target haplotype retrieved is carried by any genome in the private database. Here, the attacker could obtain the target haplotype information from the public human genomic resource like 1000 Genomes Project or PGP project. And the private human genomic database provide the summary genomic data to the attacker, either in the format of allele frequency or the format of allele existence. And by applying our method, we could either identify the known haplotypes or reconstruct the novel haplotypes. We consider our research significant because first we could infer haplotypes that is rare enough to identify one single individual from the private database. The haplotypes could have a very low prior probability and contain a very large number of variation sites. In addition, our method does not require the victim's genome, which is always required by the previous membership inference methods. And we consider two types of summary data, either the allele frequency or the allele existence. And when we consider the victim's genome is available, our method could still achieve a higher power compared to the previous ISMB-based membership inferences. Last but not least, we were able to reconstruct the novel haplotypes that are not discovered from the public resources. We note that there are previous studies related to haplotypes, however, they propose their method for a different purpose. Here I'll introduce our methods in details. Essentially, I'll introduce our methods in two aspects. The first one is the identification of a known haplotype, and the other one is the reconstruction of novel haplotypes. For the identification of known haplotypes, we consider the haplotypes that can be obtained from the public resource and to evaluate if the targeted haplotype is carried by the genome in the database. And here we propose two methods, the KHF inference for frequency table and the KHB inference for Beacon table. For the reconstruction of novel haplotypes, we consider the haplotypes that are not discovered from the public resources, and we want to reconstruct such novel haplotypes and test if they are in the database. And here we propose the NHF inference for the frequency table. We perform a likelihood ratio test LRT to identify the known haplotypes, where we set the null hypothesis as the haplotype A is not in the target database, and the alternative hypothesis is the haplotype A is in the target database. And we construct a test statistic based on the Bayesian theorem. For KHF inference, we solve the linear equations to obtain the frequency of each known haplotype fxi. As shown in this toy example, we have three known haplotypes x1 to x3 and four variation sites y1 to y4, and we have the input values as the allele frequencies fy1 to fy4, and we set the bij as the allele at variation site yj in haplotype xi. So we have b11 as 1 or 0. 1 means it is the minor allele and 0 means it is the major allele. So we know that at variation site y1, we have the frequency of y1 as the fx1 plus fx2, because only these two haplotypes contains the minor allele. And we also know that the summation of the frequency of all the haplotypes should be equal to 1. There could be three conditions found in the linear equations. If there is no solution, it indicates that the linear equations cannot be satisfied and there are novel haplotypes present in the haploblock, and we will further perform the NHF inference, which I'll talk about later. And if there is a unique solution, it means that all the haplotypes XI with a frequency larger than 0 must be in the database, and all the rest of haplotypes with frequency equals to 0 must be not in the database. And if we found multiple solutions, we cannot confirm which one is the correct one. So we use the multinomial distribution to calculate the probability of observing the input values given the haplotype A in or not in the database. For the KHB inference, we consider all the possible haplotypes that follow a multinomial distribution. We first apply the logical constraint to determine the presence of a haplotype. If an SMVC not in the database, any haplotype containing such SMV must be absent. If an SMVC is in the database, and if only one haplotype contains such SMV, then this haplotype must be in the database. Or if two or more haplotypes contain such SMV, one of them must be in the database. And for those haplotypes we cannot determine the existence, we further compute the CDF of the multinomial distribution to obtain the posterior probability and the test statistic. We know that it is quite tedious to compute the exact multinomial CDF, so we approximate the CDF by sampling according to the estimated frequency of each known haplotype. In practice, we sampled 5,000 data points in each haploblock, and it only took a few seconds to compute one CDF in a single CPU. Like mentioned earlier in the frequency table, when there is no solution found from the linear equations, we consider there are unknown haplotypes in this block, and we will further perform the NHF inference to reconstruct such novel haplotypes. Here we propose the integer linear programming approach to solve this problem. Like shown in this table, we add one more row to indicate there are D novel haplotypes x1 new to xd new. CMJ indicate the allele information at each variation site yj on each novel haplotype xm new. When CMJ equals 1, it means it is a minor allele, otherwise it is a major allele. We set the objective function to minimize the summation of CMJ over all the novel haplotype on all the variation sites. And we want to find the optimal solution to satisfy all these equations, where Km is the number of novel haplotype X and nu. We want to find the minimum value of Km since it is a novel haplotype and it is not found from the public resource. And we also want to find the minimum value of D to satisfy this equation, because we don't expect many novel haplotypes happen in one single haploblock because of the big data collection nowadays. Here is the toy example of NHF inference. We first add one more row to indicate there is only one novel haplotypes in this block, X1 nu. And we try to find the optimal solution of the integer linear programming. If there's no solution can satisfy the equations, we will add one more row to indicate there are two novel haplotypes. We will add the number of novel haplotypes until we find the optimal solution that satisfies all the equations. Here I'll talk about the experiments and results to illustrate the performance of our methods. For the identification of known haplotypes, we first built the private database by selecting 500 individuals from 1,000 Genomes project, and then we run the haploview to obtain the target rare haplotypes with a prior probability smaller than 0.1 and the number of variation sites from 50 to 187. And in total, we tested our methods on 1,350 haplotypes over 100 haploblocks, and we further built the non-distribution over the other 200 individuals to compute the test statistic of the hypothesis test. Specifically, for KHF inference, we completed experiments on 503 haplotypes over 89 blocks and were able to identify all these haplotypes with p-value smaller than 0.05. And for KHB inference, we completed all the experiment cases and were able to identify 644 haplotypes. We know that we were not able to complete all the experiments in the frequency table because we set the time restriction to solve the linear equations. So when there are too many multiple solutions, we were not able to complete the experiments. But if we are given more time, we could identify more haplotypes based on current result. Here we show the comparison between the haplotypes prior and posterior probability. The x-axis is the haplotypes prior probability and y-axis is the posterior probability. And the red dashed line here indicates the haplotype has the same prior and posterior probability. And we could see by applying our method, we could significantly improve the probability of a rare haplotype to be in the database, and in some cases we could even determine the existence of the rare haplotype. When the victim's genome is available, we compare our methods to the ISMB-based membership inference by comparing the power in each haploblock. Here we show the log ratio of our method over ISMB-based membership inference. So any cases with a value larger than 0 means our methods are better. In practical, for KHF inference, we outperformed on 82 out of 100 experiment cases. For KHB inference, we outperformed on 89 out of 100 experiment cases. For the reconstruction of a novel haplotype, we first selected haploblocks containing at least one haplotype with a frequency smaller than 0.2, and then we built the database by removing the genomes with least frequent haplotypes in each haploblock, and recorded the novel haplotypes reconstructed from the integer linear programming, and then performed the KHF inference to further confirm if the novel haplotype reconstructed is in the database. We performed the experiment considering 1 and 2 novel haplotypes are missing. We were able to reconstruct multiple experiment cases from integer linear programming, but can only report 30% of the novel haplotypes in the database by performing the KHF inference. However, all the haplotypes reported by KHF inference are correctly constructed because the integer linear programming only give you the optimal solution, while the KHF inference can confirm the existence of the haplotype. In conclusion, by applying our method, we can infer or reconstruct very rare haplotypes that are unique enough to identify one single individual. In particular, they are with frequency smaller than 10 to the minus 4 and the number of variation size larger than 50. More importantly, we don't need the victim's genome as the prior information to perform the membership inference. Even with the victim's genome available, we can achieve a higher power compared to the previously mentioned ISMB-based membership inference. In the future, we will further investigate the reconstruction of novel haplotypes in the beacon table and also the mitigation methods against the haplotype-based membership inference. And thank you for your listening.